Actually, my name is Eammon Magner. Sheenon could not be here today, and I'm representing the University of Kentucky. Today I'll be talking about a new method we have developed to query transcripts against thousands of RNA sequencing data sets. Many of you may be familiar with the Sequence Read Archive, or SRA. This is a public repository of RNA sequence data that researchers can use to save both time and money during their experiments by repurposing old data to gain new insights. Recently, the size of the SRA has been growing rapidly, almost doubling every year for the past several years. And about 2 weeks ago, there's actually 12 petabases of RNA sequencing data, which is gigantic. Unfortunately, even though we have this vast resource, it's been somewhat underutilized until recently. This is because the types of queries researchers could perform against the data was limited. Originally, we could only do a metadata-based search, so we could find samples that we are interested in, but we would have to devote a significant amount of time and computational resources to re-download and reanalyze this data to answer our queries. A major improvement came with the invention of SRA BLAST, which was the first time we could perform what is called a sequence containment query. Or given a transcript, we're interested in whether this transcript appears in some sequencing data set. Unfortunately, this method could not scale to thousands of data sets, and maybe most importantly, it could not detect novel transcripts, which is important from a biological standpoint. We can break this sequence containment query down into a few simple steps. First, we must realize that the raw RNA sequencing data can be simply represented by a list of its constituent k-mers. Now, if we break our transcript query down into its constituent k-mers, if the proportion of these k-mers which have a match in this list is greater than some cutoff, which generally the user supplies, we can conclude that this transcript is in fact present in the sample. Now, the first method that allowed us to do this with thousands of data sets was called a sequence bloom tree. This is a binary tree where every node is a bloom filter. Now, what the bloom filter does is it stores a set of k-mers in a bit vector, and it allows us to quickly determine whether a k-mer is present in this set simply through hashing. Another thing it does is it allows us to compress the data. The leaves of this tree represent the sequencing experiments themselves, and many of these sets of k-mers can be up to 10 gigabytes in size. However, using the bloom filter, we can reduce this down to only 239 megabytes, significantly reducing the size of our tree and improving performance. The internal nodes, shown here in purple, represent the union of bit vectors in all of the descendant experiments. So when we query this tree, we take the k-mers in our transcript and hash them into the bloom filter. If the number of k-mers present in the bloom filter is less than our cutoff threshold, then we can prune this entire subtree. And this prevents us from having to sequentially search every experiment to see whether the transcript is present. And this is what made this new method so good, so fast, is that we did not have to look at every experiment. Unfortunately, the original algorithm was hampered by the fact that oftentimes we would have to look at a large percentage of the tree. This was improved upon by two new methods, split sequence bloom tree and sequence bloom tree awesome. And what they did was introduce an additional bloom filter into the internal nodes. Now, instead of storing the union of descendant experiments, this bloom filter stored the intersection. Now, what this allowed us to do, we would keep a counter of how many k-mers appeared in these experiments. If at some point in our search, we realized that this counter was above our cutoff threshold, then we automatically knew that the transcript was present in these experiments, so we could again prune this subtree during our search. However, there's an issue with this. Because we do not visit the leaves of the tree, we have no way of knowing which k-mers are present in which samples. So when we query the sequence bloom tree, it only tells us which samples our transcript could occur in. And this is highly dependent on our theta parameter. As we change theta, we can get different answers. And if we want to know further information about these samples, we would have to re-download them and reprocess them on our own. Now, a natural question is, what is a good value for theta? And the answer is it's hard to tell. It depends on several things, such as how many k-mers are in the index, how long is our transcript, and what type of accuracy we would like to accept. What we would really like to answer is what we call the sequence coverage query. In other words, given the query transcript, we would like to know which of the k-mers in this transcript are present in which samples. And this allows us to do several things and gain a deeper biological meaning, such as being able to differentiate between alternative isoforms, detect mutations, and determine regions that possibly contain noise. So let's look at a quick example of how we could use a map such as this. So let's say we are given this query transcript. Notice that the k-mer coverage is actually extremely high in every sample, greater than 95% in every case. Now, if we were to query SBT, if our theta value was not above 95, 96, or 97, it would tell us that this transcript was in fact present in these samples. However, because we have this map, we can glean some further information. Now notice these dips here represent that these k-mers are actually missing. And these are actually junction k-mers that are specific to this transcript. And this tells us that, in reality, this transcript is not in fact present in these samples. Another thing that's important to notice is that we do not need any prior knowledge of a cutoff threshold to get this information. So, how do we get this information? Well, we can think of the SRA database as one gigantic bit matrix, where the rows represent the union of all k-mers present in any sample in the SRA, and the columns represent the sequencing experiments themselves. Now, for this row, a 1 means that k-mer AAAGT is present in this sequencing experiment, and a 0 means that it is absent. So if we would like to be able to answer the sequence coverage query, we need to quickly recover these rows for all of the k-mers that are present in our query transcript. Now, the issue is that we would like to query thousands of samples, 2, 3, 5, 10,000 samples maybe in the future, and we could potentially have billions of k-mers. Now, this tells us that using a simple hash map is infeasible and would take likely 100 gigabytes of RAM. This is infeasible for us because we would like researchers to be able to deploy this software on their personal laptops, which usually have only 16 gigabytes of memory. Now, when we are pre-processing the data used to test SeqOthello, we noticed something quite interesting, and that was approximately 90% of the k-mers occurred in less than 1% of the samples. And furthermore, over 50% of the k-mers only appeared in one sample. So that tells us that this extremely large bit matrix is actually really sparse. And SeqOthello is in a position to capitalize on this sparsity. We are actually able to compress this bit matrix using partitioning. So how we partition is based on the occurrence of the k-mers, the frequency of their occurrence. So we divide them into several frequency buckets. These k-mers, or this bucket would store k-mers which appeared in only one sample. And this bucket would store k-mers which appeared in exactly 5 samples. What this allows us to do is compress these k-mers horizontally. What we want to do is only store, for k-mers with low frequency, only store the samples in which they are present, and completely ignore the samples for which they are not present. Now, for k-mers with a very high frequency, we can do the complete opposite, and only store information on which samples they are absent from. Furthermore, because we only store the occurrence maps once, if k-mers share similar occurrence maps, we do not have to store this multiple times, like what would happen in the SRA database. One more thing, it also improves search locality. It has been shown that k-mers which appear in the same transcript often co-occur in similar experiments. So when we go to retrieve these occurrence maps, we will only have to load a few of these buckets into memory at a time, as opposed to having to loading several, and this will improve our query performance. Now, as I mentioned earlier, a regular hash map simply will not work for this approach. So we developed a novel two-tiered indexing structure. When we take the k-mers from our transcript query, we first map them to their respective frequency bin. And then after we have mapped them to their frequency bin, we then map them to their respective occurrence map, and this will produce our final output. Now, the speed of SeqOthello relies on the ability to do this extremely quickly in order to compete with methods like SBT. To do this, we implement a novel hashing classifier, which we call LOthello. Now, LOthello stores two hashing functions and two arrays of L-bit integers, which is where we get the name LOthello. Now, taking a k-mer as an input, each hash function computes an index into its L-bit integer array. And then the classification of this k-mer is taken as the exclusive OR of these two integers. Now, what's important to notice here is that this can all be done in constant time, requiring only two hash computations, two memory accesses, and some bitwise arithmetic to perform this exclusive OR. And this is what makes SeqOthello so fast. Just a high-level overview of what SeqOthello can do. We take the k-mers from a query transcript, and similar to SBT, we can perform the sequence containment query. We know what percentage of the k-mers are present in these samples simply by adding up the ones in each column. However, notice that we also can do the sequence coverage query because we know exactly which k-mers appear in the samples, so we can gain that deeper biological information that we would like. Now, we tested SeqOthello on the same data set that was used in the sequence bloom tree, split sequence bloom tree, and SBT Awesome papers. This is a sampling of the SRA using 2,600 samples taken from human brain, blood, and breast tissue. And our query transcripts were taken from GenCode v25. Maybe one thing that's interesting to note is, even while the size of the raw sequencing files grow extremely large, up to 10 gigabytes, the number of unique k-mers is actually capped right here. So just to go into a little bit of housekeeping details as to how we performed our testing, we used the same exact k-mer lists that were used in split sequence bloom tree. For SBT also, they had some bloom filters that we could pre-download to use for testing. Everything was performed under the Linux operating system with 32 cores and 512 gigabytes of RAM. First, we can talk about the accuracy with an ROC curve. This was done on a sampling of the 2,600 experiments. Basically, we queried 34,000 transcripts representing the entire gene set. Positive transcripts were taken using Sailfish. We estimated the TPM, and if it was over 100, this was taken as a positive transcript. And negative transcripts were taken as being less than 1 TPM. We can notice that accuracy remained essentially the same across all methods. SeqOthello did a little bit better with respect to specificity. However, an important thing to notice is that using the SBT-based methods, as our minimum cutoff approaches 1, the sensitivity that we can reach, or the number of true positives we can recover, drops significantly. Now, if we want to get a good recovery of true positives, we have to use a theta value of around 0.5. Unfortunately for SBT, this actually gives us the longest query time. So that's a trade-off we must take into consideration that we don't have to take into consideration using SeqOthello. These are some more ROC curves. We changed the ground truth to where positive transcripts were taken as being greater than 500 and greater than 1,000 TPM respectively, and the trends generally stay the same. Now we can compare how long it actually took to construct the data structure. SeqOthello was superior with respect to build time, 1 order of magnitude greater than split sequence bloom tree, somewhat better than sequence bloom tree Awesome. It took less disk space. However, split sequence bloom tree was better than us with respect to peak memory and overall index size. However, these differences are somewhat marginal, and as we mentioned earlier, our only goal is that researchers can use this on a personal laptop. So as long as these numbers are within 16 gigabytes, we're happy. Now let's look at the actual query performance. Here's where SeqOthello really shines. Notice that to query approximately 34,000 transcripts took only 2.3 minutes and 4.5 gigabytes of memory. Sequence Bloom Tree Awesome and Split Sequence Bloom Tree took much longer. Here we have 140 minutes, 220 minutes, just under an hour here. And we do have some very quick times for theta value of 1. However, it is very rare that 100% of the k-mers in a transcript would be present in a sequencing experiment. So when we use a theta value of 1, generally the search does not proceed past the first level of the sequence Bloom tree. So the search ends very quickly, which leads to these fast times. Additionally, split sequence Bloom tree does outperform us with respect to memory, but I think we can say that the query time is much more important when we're doing it in 2 orders of magnitude faster. Now we're going to look at the construction performance for the total 2,000 transcript data set, or 2,000 experiment data set. Let's go back. So the preparation time was much shorter. The index size was not smaller, however. Still is within that 16 gigabyte range, and we do believe that we can improve this further. That's something we're working on currently. For the query time, we had a much better improvement over SBT Awesome. This took a little bit over an hour. Using multi-threaded approach, we could do this in just under 30 minutes. Multi-thread approaches were not available for this software, but split sequence Bloom tree took a little over 2 days to perform these queries. So in conclusion, we are able to gain some more deeper biological meaning because we can do show k-mer coverage as opposed to just k-mer containment. We use a novel 2-tiered indexing structure, which is great for compression and is what makes SeqOthello so fast. Again, we have implemented multi-threads, and this comes with the low memory cost. We really hope that maybe in the future you all use our software in your research. I'd like to thank my partners back at the University of Kentucky, the National Science Foundation, and the National Institutes of Health. And with that, again, I thank you all for listening, and I'll open it up for questions.
We have time for one question.
There was one slide where I think you mentioned that you combined 2 hash functions with XOR.
Yes.
Maybe I missed something there, but I can't tell how that's different from just using a single hash function.
So we actually, let's see.
Oh, great talk, by the way. Looks really cool.
Thank you. Thank you. I appreciate it. We take the and/or of these 2. These aren't sequential integers, so this isn't always 1, 2, 3, 4. Actually, when this index is constructed, these could be any integers in the L-bit range. So we'll take the and or exclusive or of these 2 integers. I'm not sure if that answers your question. Actually, I didn't develop this part of the structure. My partner did, so if that was something you were interested in, I could probably get a better answer for you than what I just provided.
Okay. That's great, thanks.
So my question was actually, how do you pick the values in that table? So
No.
I guess same answer.
Yeah, this was just a toy example.
Yeah. Okay, thank you.
Thank you, and let's move on.